So the regeneration potential over as you age declines significantly. Cells can alter with age and become less efficient with age. If you look at stem cell numbers in various organs, the newborn, when you're just born, you have the highest number of stem cells, and as you age, by age 30, you get like a 96% decline in your stem cells in various organs. So by the time you're 60, pretty much the stem cell pool in various organs is really depleted. So you don't have much stem cells left in any of the organs to isolate. And plus at the same time, the stem cells can also be affected by various processes that are going on in the body because of the advanced age. And that has led to something called the theory of stem cell aging. So this basically tells you that this theory basically points to the fact that the greatest kind of stem cells in the heart or in any organ basically lose their ability to replenish themselves in tissue, and as a consequence are unable to support tissue homeostasis or tissue turnover, and so your every tissue or every organ function gets affected. A number of different things have been shown, and this is a really heavy slide that kind of gives you an idea of what happens with an aged stem cell. A lot of different things change. You have accumulation of toxic chemicals and metabolites in the cell, leads to DNA damage and epigenetic alterations that can also reduce the cell renewal ability, induce senescence and apoptosis in the cell. That can lead to accumulation of waste damaged protein. Can also prevent or reduce mitochondrial function in the cell. So there's a lot of different things that can happen in an aged cell and an aged stem cell that can reduce the overall functioning of that cell type. So before I kind of show you what we did with aged stem cells, I want to bring in this concept of cardiac stem cells and as I said before, that some of our work and what we have done in the recent past has shown that the heart itself has a number of stem cell populations, and these stem cells can reside in the atria, present all over in the ventricle and the right ventricle. A number of different cell types have been categorized. You have 3 or 4 different stem cell types which are present in the heart, and you can actually isolate those stem cell types. These stem cells, what we call them as cardiac progenitor cells, have the ability to form endothelial cell types, with muscle cell types, and then cardiomyocytes, which is the functioning cell of the heart. So what we have done over the years is that we've isolated these cardiac progenitor cells from the heart, and engineered them or genetically modified them with a protein that is called as PIM1 to make them more efficient for cardiac repair. So we grow them out in the dishes and expand them and modify them with PIM1. We create left anterior descending artery ligation in the heart, which creates a big scar tissue, and then transplant these cells back in the heart once they are modified. The animals are then followed for a period of 20 weeks, and we look at echocardiography-based assessment of cardiac function, and we showed here that the cells that were engineered with PIM1 increased ejection fraction and fraction shortening compared to the cells that were not engineered with the protein. And all of this was significant. There was significant improvement in cardiac function compared to the control animal cells. normal saline or medical. So the big question is now, whether age has anything to do with cardiac progenitor cell function. And to answer that, we kind of collaborated with the hospital and isolated these cardiac progenitor cells from patients who got left ventricular assist device implantation. So these were really, patients who had sort of end-stage cardiac failure were coming in LVAD or LVAD transplantation. And when what happens during the procedure is that when you have the LVAD going in, the surgeon actually goes in and removes the chunk of the left ventricle, and that live tissue is then sent to us, and we can isolate cardiac progenitors from that heart. So we collected overall 55 patient samples and isolated the cells, and the cells somewhat look like this, and we kind of made sure that they were stem cells and looked at various stem cell markers. And this gives you a little bit of an idea of what the patient characteristics were. We had like all different ages here. We had an 82-year-old patient, and then also a 42-year-old patient who had a really bad ejection fraction. That also kind of gives you an idea that whatever environmental conditions or patient characteristics you have can actually change the outcome of the stem cell that you're isolating from that tissue. So when we looked at various parameters for these stem cells, and we kind of isolated these cells from all these different patients, and we found that all of these cells are different. Not one cell is similar to the other in terms of how they grow. They all have different growth rates depending on what the age of the patient is, or what are some of the clinical features of the patient, and what or how advanced heart failure is. And based upon that, you can see here that the one cell type was really slow growing. And this guy here, this patient here had a really slow growing stem cell type compared to all the other stem cells that were isolated from different patients. So when we look at their telomere lengths, and you would know that telomeres are basically an indicator for aging or a marker for aging. And we found that the patient that had the slowest growing stem cell had the most shortest telomere length, which kind of also tells you that these cells have aged, and that's why they have like a really small telomere, and they have the ability to grow. So again, as I said, depending upon what the patient characteristics are, the cells can behave really differently. So going forward, we also characterized some of the cell cycle genes in the patients, and we found that some of the, again, the patient that had the slowest growing cell had also the expression of some of the classical cell cycle inhibitor genes, such as P53 and P16. And by the way, lower the bar, higher the expression, so it's a little bit confusing. So lower the bar is higher expression, and higher the bar is lower expression. So it's the opposite. So this bar here is lowest, and this means that the P53 and P16 expression is the highest in the slow growing cell. So, which again, tells you that a patient that had the slowest growing cell type had the slowest or the shortest telomeres and had a high expression of cell cycle inhibitor markers. So again, telling you that the cell environment or aging or age of the patient and the associated clinical features can really regulate how the stem cell will function in the end. So to conclude everything, you have an aged stem cell, and that aged stem cell can really have a reduced potential to repair or has a reduced potential to do any kind of degeneration. And over the period of the last few years or so, there has been a lot of different approaches to enhance the function of these age stem cells. And I've shown one of the examples of how you can actually rejuvenate these age stem cells, right? People have actually used hypoxia treatment, and you can treat condition with low oxygen therapy, and that is also improves age function. You can actually do calorie restriction and prevent overall senescence in the cell, and by reducing the calorie intake in the stem cells as well as the patient. And this strategy actually has been shown to increase aging overall by 20% in animals and in patients as well. So you can also take these same stem cells and engineer them with various proteins and growth factors to make them function better, and that way you can enhance their ability to repair any kind of tissue and in our case, repair the heart. And there are some other strategies that also include the use of biomaterials or some other patches to actually combine these age stem cells with these techniques to, again, the final goal is to rejuvenate the donor population, and by rejuvenating this population, you can actually reduce apoptosis in the heart, increase the proliferation ability of the cell, and that will ultimately lead to a smaller heart size. And I hope that in not too distant future, we may have something like this one forward that we can get some kind of stem cell-based pills to increase or rejuvenate aging or regain youth. So with that, I think I'll end here, and I would like to thank all the lab members as well as the collaborating faculty in Temple and outside in the US as well as my finding sources. Thank you all for your attention. We have a good day.